Welcome to Chapter 3.3 on meiosis. All right, so before we talk about meiosis, let's go back and review the process of mitosis. All right, and so for my students, I've asked you to do 2 drawings for interphase and to do 2 little synopsis of what's going on here. So in interphase, we're going to see the G1 phase, okay? And in the G1 phase, we've got normal cell growth and normal cell function, so producing proteins, doing its job, etc. We have a nucleus present, and our DNA is in the form of chromatin. Now, interphase is also made up of the S and G2 phases. So in these parts of interphase, we're still going to have a nucleus and we're still going to have chromatin. But in this S phase, we're going to get DNA replication. And I've already failed at fitting in these boxes. Great. So all of this is going to replicate. And here we're getting ready to divide. All right, a cell in prophase is going to look a tad different. So first I'm going to start to see the nucleus disappear. So I'm going to draw it as a line like that. And I'm also going to see for the first time the appearance of chromosomes. The spindle fibers are starting to form from the centrioles. And in early prophase, we get something like this. So we get chromosomes form for the first time. We get a nucleus that disappears or dissolves, so that membrane breaks down, and the spindle fibers start to form. All right, so in metaphase, our nucleus is completely gone, and our chromosomes are lined up along the equator of the cell. The spindle fibers are going to attach to the centromeres that are holding the sister chromatids together. So chromosomes, I swear that says chromosomes, line up in the middle. It will never say middle on an IB exam. It'll say something tricky like equator, okay, just so you know. And then the spindle fibers, which are made of microtubules, attach to the centromeres, which, as you already know, are those little things that attach the sister chromatids together. All right, in anaphase, what's going to happen is these spindle fibers are going to shorten, and they're going to bring those sister chromatids with them, okay? So they're going to get pulled apart. So the spindle fibers shorten, they literally shrink, and they pull the sister chromatids apart. All right, now it's important to note that telophase and cytokinesis kind of happen simultaneously, but we're going to draw them separately. So in telophase, the nuclear membranes are starting to reform, and the chromosomes are starting to unwind back into chromatin. Of course, I should probably draw the right number of them. So we're starting to get chromatin once again. All right, so the nuclei reform, and the chromosomes uncoil back into chromatin. And you can start to see where each of these 2 new cells is going to be. All right, in cytokinesis, a contractile ring, if we're in an animal cell, is going to pinch the 2 cells apart. So a contractile ring has formed here in the middle and literally pinched those cells apart. So here the cells separate. Again, if you are in an animal cell, that involves a contractile ring. If it's a plant cell, that involves the forming of a cell plate in the middle that'll eventually become the new cell wall. Now, mitosis, which we just reviewed, is something that we use to copy a somatic cell, like a body cell. Gametes, on the other hand, are sex cells, like eggs or sperm or pollen, have to cut the chromosome number down in half. So we just produce genetically identical cells with the same number of chromosomes as we started with. In meiosis, we have to cut the number of chromosomes in half. Okay, and that's because if we're making gametes, they can't have all of the right numbers of chromosomes. So we know that in humans, we're supposed to have 46 chromosomes in our normal cell. Well, humans are formed when an egg is fertilized by a sperm. In order for that resulting zygote to have the correct number of 46, each gamete, the sperm and the egg, can only have 23. So meiosis is the process that's used to cut that chromosome number down in half. And it does that by copying the DNA once, but undergoing not one but two cell divisions. Now, mitosis produces what we call 2N cells, or diploid. And that means that chromosomes are always present in homologous pairs, like one from the mom, one from the dad. In humans, the 2N number is 46. Okay, so N is number of chromosomes, 2 because they come in pairs. Okay, so there's 23 different chromosomes multiplied by 2 gives me my 46. Meiosis, on the other hand, produces what we call haploid cells, and that's just N. Okay, so that's where the chromosomes don't come in pairs, and in humans, our N number is 23. There are 23 different types of chromosomes. And again, this happens because in meiosis, the DNA is getting copied once, but the cell undergoes 2 cell divisions. So that is in order to produce, again, gametes, like the sperm or the egg. So in females, meiosis is going to happen in the ovary, and in males, it's going to happen in the testes. All right, now some funky stuff is going to happen to the chromosomes in meiosis. So let's back up and review a little bit. Chromosomes are going to form during prophase. However, the DNA is replicated prior to that, and of course that happens in the S phase of interphase. We went through that earlier. Okay, and that's why most chromosomes have this X-shaped structure. So again, when chromosomes are going to copy themselves, they're at first present in homologous pairs. So one from the dad and one from the mom. When they copy themselves, the other copy is going to attach to the original copy via a structure called the centromere. So these are what we call a homologous pair. They include the same genes in the same location. You should know that by now. Okay, it's just that one is from the mom and one is from the dad. Okay, so this one would be maternal, and this one would be paternal from the dad. All right, now these structures, each half here is of course the sister chromatid. It's a result of replication, so each sister chromatid is identical to the other one, and they are held together by the centromere. All right, so again, in meiosis, we're copying the DNA once, but we're dividing twice. And so it's basically going to go through all those phases that we talked about with mitosis twice, except for interphase, only going through interphase once because we're only copying the DNA once. So we're going to go interphase, prophase, metaphase, anaphase, and then start another round of cell division. And since we go through them twice, you're going to see me use these Roman numerals 1 and 2 to delineate between the stages. All right, so we know during interphase we're going to get DNA replication. So let's talk about prophase. In prophase 1, we're going to see the condensation of chromosomes. And in prophase 1 of meiosis, something crazy called crossing over occurs. And crossing over only happens with homologous chromosomes. Remember, they have the same genes in the same location. So a pair of homologous chromosomes, again, one from the dad and one from the mom, are going to kind of snuggle up to one another. And when they do that, they are going to exchange parts of their chromosomes, kind of like exchanging trading cards, right? So they're going to switch those sections of their chromosomes. And that's okay, right? Because if this is the gene for eye color and this is the gene for eye color, all they've done is swapped those. It's still the same genes. And maybe only different versions of those genes. Okay, now the number of spots where they cross over is totally random, and so is how much of that genetic material is exchanged, okay? So this can result in what we call recombinance, okay? We're recombining genetic material. So before, okay, I had these two sister chromatids, one here and one here, and they were identical. And now after crossing over, those sister chromatids are no longer identical. They're different. So we're recombining things. All right, and this is called crossing over again, and the points where these cross over are called chiasma or chiasmata. And those, again, are the spots here where they cross over. When crossing over occurs and these sister chromatids eventually split into different gametes, so this one ended up in this gamete and this one ended up, let's see, down here, okay, it ensures that the gametes that I produce eventually through meiosis are different. And so what that's going to do is it's going to give our potential offspring, so any offspring resulting from this and resulting from this are going to be different. It gives us quite a lot of genetic variation, and that's a real advantage for sexually reproducing organisms because it ensures that if there's some kind of a change in the environment, like, I don't know, a bird that likes to eat white bunnies, okay, it ensures that maybe some of our other offspring are going to have different genes in order to survive those changes. Now, after prophase comes metaphase, and we know that in metaphase we're going to line up these chromosomes in the middle. But what's interesting about metaphase 1 of meiosis is that they're still in their homologous pairs, their buddy that they just snuggled up to and swapped information with. So these homologous pairs are going to line up in the middle. This is metaphase. This is what metaphase looks like. It's okay for them to be stuck together like that as long as they're lined up, okay, in the middle. Now, they don't line up according to any type of rules. So this homologous pair could line up like this, and this homologous pair could line up like this, where both of these orange ones are on the left-hand side, or they could line up like this and this, where now the orange is on the left and orange is on the right and the blue is on the left. They can flip-flop. So could have these, okay? So the order in which they line up is completely random, and it's called random orientation. And so if we think about what's going to happen with our gametes, okay, eventually this is going to become a cell, this is going to become another one, and then those gametes are going to split. This one's going to split from this one. Okay, you can see how lining up differently results in different combinations of genetic material. So again, it's another way that we increase the variation in those gametes. All right, so let's talk about the steps of meiosis. So if you're doing notes for me, you've got a big long diagram there with all these stages on there. I want you to annotate them with what's going on in each phase. So in interphase, again, interphase are really long. We've got the G1 phase, which we're doing growth and normal cell function. We've got the S phase, so we're going to replicate that DNA. And we've got the G2 phase, preparation for division, production of enzymes, etc. In prophase 1, okay, so again, we've got chromosome formation, and then we're going to have crossing over. So they're going to exchange that genetic material. We just talked about that. And just like in regular mitosis, those spindle fibers are going to form, and we're also going to see the nucleus dissolve. So that nuclear membrane here is going to start to disappear. In metaphase 1, we're going to line them up in homologous pairs, okay, and they line up randomly in the middle. Again, that's called random orientation. So I have them shown like this, but they could have easily just have lined up in a different orientation, flipping sides or what have you. Okay, in anaphase 1, these spindle fibers that were attached to the chromosomes are going to start to shorten, and they're pulling those homologous pairs apart. So notice that the sister chromatids are still connected. Okay, we didn't pull that X shape apart. They're still connected. What's being separated are the homologous pairs. So these two blue ones were homologous pairs. They were kind of stuck together after crossing over. And in anaphase 1, we've started to pull them apart. All right, so after we've pulled them apart in anaphase, okay, we're going to move into telophase, where that nucleus starts to reform. The spindle disintegrates because it's already done its job, and these chromosomes are going to uncoil back into chromatin. So notice that these are not in homologous pairs anymore. So this guy's homologous pair is now over here, and we're about to separate their cells. And since crossing over has occurred, okay, the two halves of that chromosome are no longer identical. Okay, one of them has exchanged a bit of information, and in a moment, when we split them during meiosis II, you're going to notice that one half is not the same as the other. Again, they contain the same genes, just not the same alleles. All right, now we're going to skip interphase because we don't need it, all right, because those cells aren't replicating their DNA. So right after we split them, we're going to move directly into prophase II. You're going to notice that prophase II, metaphase II, all of meiosis II looks very similar to what's happening in mitosis. Okay, so in prophase II, that DNA is going to condense into chromosomes. Okay, the spindle fibers form. I keep forgetting to write that the nucleus dissolves. Okay, so we are starting with the two cells that we had from meiosis I. Okay, and in each of them, we're going to see prophase II take over. Okay, nuclear membrane dissolving and chromosomes are forming. In metaphase II, this is going to look almost exactly like mitosis. So in each cell, the chromosomes are lining up in, again, a random orientation. So notice here that this chromosome, the part that had experienced crossing over, lined up on the left, but it could have easily just flipped around and lined up with that part on the right. In anaphase II, these spindle fibers are going to shorten and pull those sister chromatids apart. So we're kind of getting rid of this X shape. Okay, we're pulling those sister chromatids apart, and you can begin to see where each new cell is going to form. All right, I want you to take a hard look at what's going on in telophase II. It's not that telophase II is complicated. Same thing as in mitosis, right? So reforming the nuclear envelope, that's going to uncoil back into chromosome. But I want you to notice that we're going to have 1, 2, 3, 4 cells, and that each one of them is completely different. So again, they're haploid. They don't have a homologous pair. It's just from the mom or just from the dad that the little chromosome doesn't have a pair, the big chromosome doesn't have a pair, and they're not identical. These cells are not identical. This one contains different stuff than this one, okay, so on and so forth. So we're going to produce 4 haploid cells that are all genetically unique. All right, so just to do a quick recap here, meiosis I is where all of the weird stuff happens, okay? So we have replication, but then we have crossing over, okay? And we're lining up as homologous pairs in metaphase I, and then that's what's separating in anaphase I. Meiosis II is a lot more similar to mitosis, except for that we're not replicating before. There's no crossing over, the chromosomes are lining up, and we're separating those chromatids during anaphase II. So let's ask ourselves an interesting question. Let's say I have two identical twin females, identical meaning came from the same zygote, had the same DNA. And I have two identical twin males, and if one of them has a baby with the other one, and the other twins decide to also have a baby, will their babies be identical? Well, let's take a look at the odds of that. Okay, so in order to make a baby, we've got to form gametes, right? So gametes are genetically unique, and that's because they not only cross over, but they randomly align themselves in metaphase. So we have a formula for figuring out the number of possible gametes we can make, okay? And this is not how we want to write it. It's 2 to the nth power, where n is the haploid number. So for humans, okay, that would be 2 to the n, which would be 2 to the 23rd power, because there are 23 different chromosomes. That's the haploid number. And that means that each human is able to make a whole lot of different gametes. And this number comes from the number of different ways that those chromosomes can align themselves in metaphase. What it doesn't even take into account is the variation produced by crossing over, because we can't predict how many places are going to cross over, how much exchange is going to take place, okay? So realistically, this number is a lot higher. We just don't have a way of calculating it, because we don't have a way of calculating how much crossing over can take or can account for. So something like a fruit fly, where its haploid number is 4, okay, is going to have a lot less potential gametes. So 2 to the 4th power, you can figure that out, okay? That's going to have a lot less potential gametes than a human, because we have more chromosomes that are able to randomly align. All right, so we have like all those millions of potential eggs that can be created, and all those millions of different sperm that can be created. And when fertilization takes place, the nucleus of the egg and the nucleus of the sperm are going to fuse, and that's how we get our homologous pairs, one from the mom, one from the dad. So we can produce all these 2 to the 23rd power eggs and 2 to the 23rd power sperm. The likelihood that one of those like millions of eggs and one of those millions of sperm fertilizing and both being the same and both being the same for each identical set of twins is just so astronomically small that I think it's safe to say that those offspring are not going to be identical. Now, during anaphase 1 and anaphase 2 of meiosis, we rely on those chromosomes separating, okay? So here's what's normally supposed to happen. Those sister chromatids are supposed to separate, and one is supposed to go into each new cell. Well, what happens if something goes wrong? That is called non-disjunction, okay? So junction meaning to join, dis meaning to not join, so that not join means to separate, and then non means not. So this literally means not separating or accidentally sticking together. So if chromosomes stick together, that's going to result in improper chromosome numbers in the resulting gametes. They're supposed to separate. If they don't, one of the gametes is going to have too many and the other one will have not enough. So in humans, meiosis and the separation of those chromosomes is supposed to lead to a sperm with 23 chromosomes fertilizing an egg with 23 chromosomes. Well, let's say in the egg that non-disjunction occurred and one of the eggs ended up with 22 and the other one ended up with 24. If one of those eggs with an improper chromosome number is the one to get fertilized, then the resulting zygote is going to have the incorrect number of chromosomes. So over here, we're supposed to have, again, 2 of every chromosome, one from the mom and one from the dad. Well, if these chromosomes failed to separate during meiosis to form the egg, and it's fertilized by a sperm from the dad, the resulting baby could have 3 copies of a chromosome instead of the normal 2. If that occurs on the 21st chromosome, like they stick together and the zygote has 3 copies of chromosome number 21, we call that trisomy 21, somy referring to the chromosome, tri meaning 3, 21st is the number of the chromosome, and we also call that Down syndrome. A lot of the times, if the zygote has 3 copies of a different chromosome, that can lead to such grave abnormalities that the fetus is unviable and a miscarriage will ensue. Trisomy 21 can result in a viable fetus and a healthy child, just one with Down syndrome, which is characterized by stunted growth, low muscle tone, which can impact the speech, characteristic facial features, mental impairment, a whole host of unique genetic issues. Now, we can actually figure out if a baby has Down syndrome before it's even born. And to do that, doctors need to obtain fetal cells and pluck out those chromosomes to determine if there are 3 copies of that 21st chromosome. All right, so why would a doctor guess that a baby has Down syndrome? Well, on these ultrasounds, they're looking for an abnormal amount of fluid underneath the neck that can be characteristic of Down syndrome. So in that case, the doctor is going to want to come in and it's going to want to get some cells from the fetus and again pluck out the chromosomes and see if there's an abnormality in the number of chromosomes. And there are 2 methods for obtaining these cells. What we don't want to do is stick a giant needle into the baby, okay? That could induce a pathogen. We don't want to poke the baby. We don't want to do any of that kind of stuff. So we may choose to do what's called an amniocentesis. And that's going to involve inserting a needle into the amniotic sac, which is this big water balloon the baby is growing up in, and plucking out some of the skin cells that the baby has shed throughout gestation or pregnancy. So as long as those are the baby's cells, it'll have those chromosomes and I can karyotype them. The other method is called chorionic villus sampling, and it also involves a needle, but here we're not taking them out of the amniotic sac. We're taking some cells from the fetal side of the placenta. Both of those methods are going to obtain cells that we can use to diagnose Down syndrome or a host of other genetic diseases, but they aren't without risk, okay? So they do involve an increased risk of infection, injury to the fetus, and miscarriage. However, once we have those cells, the doctor can take the DNA out and replicate it to make a bunch of copies and use it to make a karyotype. And that karyotype is going to allow us, well, actually, they would be making a karyogram. The picture is called the karyogram. The diagnosis is called the karyotype, based on any kind of chromosomal abnormalities that they may see. Okay, so over here in this karyogram, right, I'm noticing that first of all, we have a girl, 2 X chromosomes, and all the other chromosomes seem to be in normal 2s, normal pairs. Over here, in this one, so we have a baby boy, there's an X, well this big one is an X and this little one is a Y, so that's a boy. And I'm also noticing that all the chromosomes are coming in pairs except chromosome number 21. So this would be a genetically healthy baby girl, and this would be a boy with trisomy 21 or Down syndrome. Now, this graph shows the risk or incidence of Down syndrome, so the percentage of births that end up in Down syndrome, versus maternal age. So maternal age is the age of the mother at birth. And what we can see here is that maternal age is a very highly correlated variable with Down syndrome. It's the #1 risk factor for Down syndrome. So women in their 20s experience close to 0% Down syndrome births, okay, where a woman of 45 that gets pregnant has a 3.5% chance, okay? So we're talking about an exponential increase in the risk of Down syndrome through maternal age. And that's just because of the way that females form their gametes. So males produce fresh sperm every day, millions, hundreds of millions of sperm every day undergoing meiosis. Okay, in women, those oocytes, okay, are present at the time of birth. And so they're around from birth all the way up through the fertile years. So they're getting older and older and older. They're not replenished. Those oocytes sit in the ovary, and then they undergo meiosis each month. And then again during fertilization, when the woman is trying to get pregnant. So the older the woman is, the older her oocytes are, and the more likely it is that there's going to be a problem with non-disjunction, with separating. So something really interesting to take a look at there. And that'll do it for chapter 3.3 on meiosis.